Alright, ladies and gentlemen, welcome back. We're looking at part one of the cell, and we're just going to be talking about the different components of the cell and what they do. As you can see here from these two pictures, these are two different cells, and depending on what's in them and what the composition of them will determine their shape and their function. Okay, so let's get right into it here. We have two different types of cells. So the ones we're going to be focusing on are eukaryotic, which essentially means that they are membrane-bound organelles in them. So things like the nucleus and mitochondria, vesicles and vacuoles will have membranes surrounding them, whereas things in prokaryotic organisms like bacteria do not. They will only have the ribosomes and genetic information. Okay, so they also have a nucleus, and chromosomes are linear, and cells divide by mitosis. So nothing brand new for this, since you guys have probably seen this in grade 9, but let's get right into the different organelles. So the first one we know about is the nucleus, and I want you guys to know the actual responsibility of the nucleus. It contains the genetic information DNA, and the DNA has the code for the order of the amino acids. So in order to determine what a protein's function is, it has to have specific order of these amino acids, and if they're in a specific way, it'll have a specific function. Okay, so they have that code. What they also contain is that DNA is organized as chromatin, which is just this loosely interjumbled organization method, but it will condense into chromosomes, those linear shapes, once it's ready for cell division. It also contains a nucleolus, which essentially contains ribosomal RNA and ribosomal RNA and proteins, and which basically make up the ribosome subunits that we need to make those proteins. It is surrounded by a double membrane envelope, and that's the nucleus I'm talking about, not the nucleolus. And then they have pores for substances to get in and out of. And finally, when we want to change DNA into more DNA, or change DNA into RNA, like transcription, it happens inside the nucleus, which we just saw last unit in DNA. So you can pause that and get that down, and you'll have all the diagrams of the organelles in your note package. Okay, the cytoskeleton, we have kind of two parts here. The first one we're going to look at is the microfilament. So this is the part, the substances that give the cell the structure that it has. So the first one's microfilaments. It's composed of actin, which is a contractile protein. And what it allows things to do is actually move within the cell. And if you contrast to the microtubules, they're composed of tubulin, and these would be the components of centrioles, which are used to get the spindle fibers going in the process of mitosis, meiosis, and they also determine the cell structure, as you can see from this picture. Okay, so here are the little microfilaments here, and then these ones all in here are the microtubules that are essentially giving some structure and stability to those cells. Okay, so you can pause that and get that down. Ribosomes, we've talked about those last units. This is the site of protein synthesis. They're going to bind to the mRNA in order to allow the tRNA to come join and bring the amino acids. You have two different types. You have free ribosomes, and you also have bound ribosomes. The free ones will be in the cytosol or the cytoplasm, and their function is to make proteins that are going to be used by the cell. So either in the nucleus, like the helicase and DNA polymerase enzymes like that, or actually within the cell membrane. We also call them polysomes because there's usually multiple ones together, and they can actually do multiple protein synthesis at the same time. They're just carrying on one behind each other. The bound ones are on the ER, and these are the ones that are going to be producing proteins that will be bound and exported out of the cell for other uses. Okay, so jot that down. The ER, endoplasmic reticulum, we know that we have two types. It usually surrounds the nuclear envelope, nuclear membrane, which would make sense because the proteins that, or the RNA that comes out of the nucleus is going to go to the ER. First of all, the smooth ER, this one has no ribosomes, hence the name smooth. It synthesizes or produces lipids, and also metabolizes some carbohydrates. And finally, detoxes drugs and poisons. So there is a lot of smooth ER in liver cells because that is the function of the liver. If we're looking at the rough ER, this is where we have ribosomes, hence the rough, and also the synthesis of secrety proteins, and those are the proteins that will be destined for outside of the cell. Okay, so you can copy that down. The Golgi apparatus, where those secretory proteins and lipids are going to be shipped. So anything that's made in the ER, that they're going to get modified, stored, and then shipped to another destination. Essentially, what it does is it gives it the destination at the Golgi. So you're this type of protein, here's your tag, you're going to go to a specific spot. The flattened membranous sacs are called cisternae. They have transport vesicles that bud off of them, so it can take the transported proteins to the membrane. And it also creates our lysosomes. What's a lysosome, should you say? You will just have to hold on. Bam, lysosomes. So what these are going to be is it's a sack of hydrolytic enzymes, essentially proteins that use hydrolysis to break apart macromolecules. There's about 36 different enzymes that are going to be in those things, so they can break down essentially 36 different items. They use phagocytosis, which we'll talk about in the cell membrane section, which is essentially it engulfs various substances. Tay-Sachs disease is essentially a lysosomal disorder where you cannot produce lysosomes properly, and as a result, if there's no lysosomes, there's no breakdown of these like glycolipids and glycoproteins, and they build up and end up causing the neurons in the brain to die, and so this disorder can actually lead to death, which I just said. Okay, next one is going to be the peroxisome. Peroxisome is similar to the lysosomes in terms of its structure, where it's going to have these enzymes inside, but instead of breaking down the macromolecules, it's going to be breaking down toxins similar to what the smooth ER did. And it does it by taking these toxins and converting it into hydrogen peroxide, hence the perox, and then continually to convert that down into water. And it uses catalase and peroxidase in order to do that. So you can pause and get that done. Oh. And they come from the endoplasmic reticulum. Okay, vacuoles. The ones we're going to be focusing on most is going to be in the plants. The plants, essentially, it's going to be used for salt and water storage along with some other components. This is where in plants, when the vacuoles are full, that's when you're going to have these rigid leaves. And when the vacuoles are not full, that's when it's going to become wilted. Okay, and we'll give a little more detail on vacuoles in class. So pause that and get it written in. So mitochondria, now we're getting into our power organelles. This is the organelle that creates ATP through cellular respiration. We have two main parts here that you can see, the cristae and the matrix. And then we have the intermembrane space. Unique little characteristic about mitochondria is it contains its own DNA that is inherited from your mother. Okay, so this goes back to the evolution of the cell. Scientists believe that the bacteria got into the cell, and those bacteria that were engulfed ended up turning into the mitochondria. Cellular respiration, again, it's like as I mentioned, is used to create ATP. So we take glucose, so write this formula down, and oxygen will be converted into CO2 and water, and a awesome byproduct of that is ATP energy. So that's the maximum that you need to go into in terms of depth for cellular respiration. But just as a little heads up, this is where you would go eventually if you were learning about cellular respiration. This guy. It's called glycolysis and the citric acid cycle using the electron transport chain. There's a lot going on there. So take a peek if you're at all interested, but we will not be going into this depth. Okay, photosynthesis is done in the last organelle we're going to deal with, which is the chloroplast. It has a pigment called chlorophyll that's able to capture sunlight energy and convert it into usable sugars. So you can copy that stuff down here, and then we'll look at the actual process. So the process looks like this. Its whole goal is to take CO2 and water, CO2 from the atmosphere, water from the ground, and be able to use the energy from the sun to catalyze it into sugars that it can use, and oxygen. And it does produce some ATP, but almost all that ATP that's used or that's created is used during this process. And so the whole goal of this is to get the sugars, because we can go to the store and get our sugar. Plants cannot, so they have to produce their own so they can use it in the mitochondria in the cellular respiration process we just talked about. Just like cellular respiration has some craziness, so does photosynthesis. This one is a little bit in depth as well. Okay, so you guys can take a peek quick at this one just to see how in depth it goes. And that is all for us today. So we're going to go into some more detail when we get into class, but yeah, you definitely do not know all of that. Have a good night.